Okay, today we are looking at genetic engineering for insulin production. In the Year 12 book, this starts on page 35, genetic engineering. So just before we look at any diagrams, we'll give a wee introduction to genetic engineering. So genetic engineering is the taking of a piece of DNA, usually a gene, from one organism and adding the genetic material to another organism. So genetic engineering is defined as a process that modifies the genome of an organism to introduce a desirable characteristic. So in this case, the donor will be the human DNA, more specifically the gene for insulin, and the recipient will be a bacteria. So the bacteria's plasmid is going to be changed. It's going to contain the human DNA, the human gene for insulin, and then that bacteria will be able to create insulin. That's the desirable characteristic. Okay, so we're just going to talk through the steps of this. So the first step is to obtain a human cell, go into its nucleus, and cut out the gene for insulin. Now, when this gene is cut out, it will have what's known as sticky ends, and we'll talk about that at the end. So for now, all we need to know is that the insulin gene, which is found in the human chromosome, is cut out, and that is completed using enzymes. A bacteria cell is then taken. The plasmid is removed from it, which we should remember is a ring of DNA, and it's removed from the bacteria cell and prepared so that it can be changed. Now, it is cut open with the same enzymes that cut out the human insulin gene from the human DNA. And that gene is then inserted into this little gap here. The new plasmid, which contains all its previous DNA, plus this gene for insulin production, human insulin production, if we're being really specific, is now placed back into the bacteria, and then that bacteria will be placed in a fermenter. Now that fermenter will have all the conditions which allow reproduction to happen really quickly. So it'll be the right temperature, there'll be lots of nutrients, and so forth. So it will manufacture really quickly and reproduce really quickly. And as a result, we will have millions of copies of this bacteria, and this bacteria can make lots and lots of insulin. So pure insulin is then obtained by filtering it and purifying it. This process is known as downstreaming. Now, the advantages of this is that it does not cost a lot of money. It's relatively cheap. It's extremely quick. Another advantage is that it replaces the old method of treating diabetes by taking insulin from animals. So this would be a far more ethical way of doing it. Okay. A lot of people will not want to use animals, and a lot of people are afraid of the side effects of using insulin from animals as well. Now, I mentioned earlier that whenever the gene is cut out, so here it looks like it's just a rectangular section that's cut out, but I did say that there was these things known as sticky ends, and that's what we're just going to briefly talk about now. So when the enzyme cuts out the gene, it leaves these sticky ends, and when it cuts out the plasmid, cuts it open, it also leaves the same sticky ends. As a result, the gene can fit nicely into the plasmid by base pairing rules. So these bases, these pairs, will pair up, form bonds so that they can stick together, and it means that the human gene is going to actually be sort of stronger, like it's more stable within that plasmid as a result. Okay? So that is a very, very quick introduction to genetic engineering. Sort of really goes over it pretty quick. So it's important that you read your book for more information. There's a video clip here, sort of a clip here, and another video clip here, which are worth watching as well. Okay, so good luck. Any questions, please post them immediately.